Hello, my name is Elena Garralda. I lead the Early Drug Development Unit in Vall Hospital in Barcelona, Spain, and I'm currently the chair of Recist for the ORTC.
I'm Scott Laurie, and I'm a thoracic medical oncologist and the head of the Phase 1 clinical trial unit at the Ottawa Hospital Cancer Centre, and I'm a professor of medicine at the University of Ottawa in Ottawa, Canada. Together with Elena, I am co-chair of the steering committee of the Recist working group.
So as you may be aware, Recist is a working group that started in the late 90s by three main groups, the ORTC, the NCI, and CCTG in Canada. And they came together to really bring a method, a common language on how we assess response of treatment in cancer. So we know that normally tumors don't shrink on their own. So the idea was, if the tumor is shrinking, this is due to treatment response. And based on the analysis of a lot of data sets that were collected, they came to a criteria in which we were able to analyze which of the tumors are responding to a concrete treatment, which patients are stable on that treatment, or which patients are progressing upon the treatment.
The Recist working group consists of volunteer physicians and scientists from across the globe with expertise in anti-cancer drug development and clinical trials, radiology, statistics and data science, and cancer biology. Together, the Recist working group is tasked with ensuring the continuous refinement of Recist to ensure it remains the preferred tool for response assessment.
Recist has really shaped the way we currently perform drug development, because based on this criteria, we are able to say if a tumor is responding to a certain treatment or not. It has created a common language that all investigators are able to use. One of its beauties is that Recist is simple. It can be evaluated in different sites, across tumors, and across multiple trials. So this really helps understand the value of a treatment.
The main benefits of Recist for clinical trials in solid cancers is to define what constitutes a response to anti-cancer agent or agents in a standardized manner. This allows for a common language in the cancer research community and permits comparisons of different agents or treatment strategies across different clinical trials. Besides allowing for determination of rates of response, Recist permits the determination of several response-based endpoints, such as duration of response or disease control, and progression-free survival.
Now we see approvals of drugs based on Recist endpoints such as progression-free survival, which is based on progression upon Recist.
For patients enrolled in clinical trials, standardization of response assessment helps to ensure ethical conduct of trials and provides the ability to document changes in an individual's tumor burden with exposure to an investigational agent, contributing to participant protection. Recist-derived endpoints from clinical trials are also often used for submission to authorities for regulatory approval, thus helping to make new, effective therapies available to patients outside of clinical trials.
Currently, there are new technologies that perhaps can help us in the future beyond the image on the tumor to see if we can better define tumor response. So in the next years, we will be focusing on how we can improve Recist beyond the traditional criteria to see if we can still improve the way we are assessing if a tumor is responding to a certain treatment. This has impact both for patients, for the individual patients, because it can help the way the patient is managed in the clinic, and it also has a lot of implications for the way we develop drugs. If we have clear signal, if a work is working or not, we can really decide on the next steps for that drug. Is it worth pursuing the development of that drug, continuing to a later stage of development, or should we stop that drug further? So it has implications both for patients and both for all of the stakeholders that participate in drug development.